Okay, everybody, good morning. We'll go ahead and get started now to keep us relatively on time. So thanks again, everybody, for being here bright and early. This is the technology and research session. This is the 5th year that this session has happened. And as moderators, we took some inspiration from the TDN spring meeting with conversations about different biological sample types that may be used to detect early infection or used generally in the highly effective modulator era when people are less able to provide sputum. So we want this session to be something we as research coordinators can understand what might be coming at us. What will we be asked to collect, and what these biotech samples mean for the science. We'll also discuss the use of mobile health applications in CF research and explore the challenges of online surveys and how we can secure data collection. So we'll hear from Dr. Gina Hong and Dr. Manu Jain about biological samples beyond sputum. Then we'll move into a discussion about mobile health apps from Ellie Bernstein and Julie Gowdy, and how to detect bots interfering or not in survey technology from Rachel Salyer. So it's my pleasure to introduce Dr. Gina Hong. Dr. Hong is an assistant professor of medicine at the University of Pennsylvania Perelman School of Medicine and associate program director of the Penn Adult CF Clinic in Philadelphia. Her research focus is on the study of chronic infections in CF, and she is the national PI for the upcoming SEND CF study.
Thank you so much. Thanks for the applause before I even said anything. So thank you to the organizers for allowing me to present this work or present this presentation on behalf of the CF team. I'm actually pinch hitting as well, so I'm really just happy to be here. I'm going to be talking about the novel approaches to detect pulmonary infection and cystic fibrosis. So this is just the NACFC disclaimer. I realize that I should have probably disclosed that I am the national PI for the CF study, but otherwise no other relationships. So today we are going to be discussing how the landscape of infection surveillance has really changed, primarily because of changes in respiratory sampling that we've observed because of the widespread use of CFTR modulators. We see that sputum is just less available. And so it's more limited in its utility across the CF population. So in response, we really need non-sputum dependent sample types. And so we are going to explore some of those promising sample types today to improve infection detection in CF, including breath, urine, and blood-based diagnostics. And then I hope to introduce you to the CF study, which is really a study that's going to be collecting biospecimens and metadata to help support where many of us are at right now, which is really developing proof of concept and performance testing for diagnostics in this space. I think this figure was actually presented in the plenary yesterday as well, and many of you are familiar that the PROMISE micro sub-study has demonstrated that really there's been marked Pseudomonas sputum density drops after early initiation of elexacaftor, tezacaftor, ivacaftor, or ETI. And what's really remarkable is that these drops in sputum density have been sustained even close to 5 years out. But also what we observe from this group, which is published already in the Sing Lab, and these are preliminary data showing that durability within that 5-year mark, is that although sputum density has significantly decreased in people with ETI, that Pseudomonas infection continues to persist, both by way of detection from culture and also the DNA-based assays that they use. And so, although people are on modulators, these lung infections have decreased in severity but remain partially unresolved. We also know that CFTR modulators really have enhanced mucociliary clearance and also has improved the immune response in the CF airway. And we all know also in this room that chronic infections are burdensome, particularly for our patients, and will worsen lung health over time. So taken together, we really do need new diagnostics or infection detection methods to allow for this early identification, because there may be a greater potential for eradication because of the positive effect modulators have had. And so these are data from the CF Foundation Patient Registry, really depicting the real-world respiratory sampling that we've observed. So the x-axis is time, y-axis is counts, and you could see that the sputum samples that are captured by the registry are in the blue, and the throat or the oropharyngeal swabs are in the red. And we see that, because of COVID lockdown, there was this precipitous drop right after 2019. And we all also know that the end of 2019, that's when ETI was approved. And so after that point, we see this slow rise of the oropharyngeal swabs in red, but the sustained kind of lower level of the sputum in blue, which I interpret these data as that oropharyngeal swabs are really replacing the sputum for respiratory sampling, because sputum is less readily available. Patients are not able to expectorate them clinically as easily. And this is a bit problematic because oropharyngeal swabs, although we can detect infection, we know that they are less sensitive, which really threatens infection surveillance in our population and really increases the risk to miss infection. And so the Cystic Fibrosis Foundation really has responded to formulate a detection strategy to look for non-sputum dependent diagnostics, which really the hope of these diagnostics are for early detection with high sensitivity, and also hopefully can overcome some of the shortcomings of culture-based testing. And some of the three promising approaches that the foundation have identified include breath, urine, and blood-based detection methods. And at this stage, there really has been a focus for detection of Pseudomonas aeruginosa. And this is primarily because that the detection of Pseudomonas aeruginosa has a lot of treatment impacts. For example, things like the decision to eradicate or may impact the treatment for pulmonary exacerbation therapy. But we also do recognize that there's also other important CF pathogens that need to be studied and are critical to be studied in the future. So let's introduce you to some of these diagnostics. So how can breath technology diagnose respiratory infections? So within the human host, when in exhaled breath, there are these molecules called volatile organic compounds, or VOCs. These are really end products of metabolism. And what happens is that if an individual has, say, like a bacterial infection or a disease state, that these bacteria actually exude these or produce these VOCs, and these VOCs can then be captured through different mechanisms, including something called a sorbent tube, which is kind of schematically depicted there. And they can be stored and then analyzed, in which those VOCs can be identified through two main sensing methods, which include mass spectrometry and direct biosensing. So through these really analytics, that these VOCs that are produced by bacteria can be associated with that said bacteria to aid in the diagnosis of, say, infection in this case, but in also other disease states. And so this has been explored in other pulmonary diseases and actually non-pulmonary diseases, such as small intestinal bacterial overgrowth. And so the CF Foundation had recently invested in a company called Alstone Medical, which is one of the leading companies to really explore the work of breath and identifying VOCs in different disease states. And this is just a picture of their mechanism, how they collect the breath. And so this is actually collected through this kind of mouthpiece, and that I don't think I have a pointer here, but there's these like two metal tubes below on the bottom of the mouthpiece, and that's the sorbent tubes that actually collect the breath, in which that can be actually stored and also shipped over to get analyzed through mass spec. And then through then those VOCs that are produced by that bacteria can be associated with the bacteria. And there has been, again, these are not Alstone data, but there has been investigative groups that have identified particular VOCs in chronic Pseudomonas colonization in children with cystic fibrosis. So although this is very early stages, the goal, at least for Alstone, is to help identify that VOC signature or profile that can help us and aid in the diagnosis of Pseudomonas aeruginosa in CF. So with urine, there are existing urine assays that aid in the detection of pulmonary infections that we use even in our ICUs very regularly, such as Legionella urine antigen and Streptococcus pneumonia antigen. But there are other novel diagnostic methods that are being explored for the detection of infection that may be excreted in the urine. One of which on the left-hand side are extracellular vesicles, in which these extracellular vesicles, or EVs, can contain outer membrane proteins of the bacteria and then can be excreted in urine. On the right-hand side are the alkylquinolones, or AQs. These are quorum-sensing molecules, that are generated by the bacteria, and these also can be detected in the urine. And so that there are two companies on the right-hand side. Pearl Diagnostics is developing urine technology to try to, to develop a enzyme immunoassay to detect these bacteria components in the extracellular vesicles that are excreted in urine. So this is in the very early proof of concept stages, but is something that is being explored. And then on the right-hand side, a company called MyDX, which is based in the UK, have generated this proof of concept lateral flow assay that that developed an antibody to bind to these AQs and then detect them excreted in the urine. So again, these are very early stages, but show the promise that even urine can be used as a non-invasive biospecimen for Pseudomonas detection. And now blood. So I think many of us are familiar with cell-free DNA. Cell-free DNA is essentially nucleic acids that are released into the circulation and then can be detected. It's used in cancer diagnostics for tumor genetics and also prenatal screening. And there's growing work that is showing that microbial cell-free DNA can also identify infection. And so the Salaponte lab here in Seattle have demonstrated that cell-free DNA, or Pseudomonas aeruginosa cell-free DNA, is significantly greater in detection in plasma circulation compared to those with CF and without positive Pseudomonas cultures, and also compared to the non-CF host, really kind of showing the promise that these cell-free DNA components related to the bacteria can be detected in the circulation. Although this is a very small sample, they are expanding this to, I believe, other promise samples. So we recognize at this time that really there's a need for non-sputum-based methods, but there needs to be a way for us to access these CF clinical samples and really have kind of robust, well-characterized infection status associated with these samples. So there needs to be a study that also collects these novel samples, but then really captures that clinical gold standard, which currently right now is sputum. And so this is what has led to the CF Foundation formulating the SEND-CF study, which stands for the Study to Enable New Diagnostics for Pulmonary Microbes in People with CF. This is a multi-site cross-sectional specimen collection study, which the goal is to collect and bank biospecimens of people with CF and specific microbiologic profiles and make them available to the wider research community to test and validate the diagnostic assays for the detection of relevant CF pathogens with that focus on Pseudomonas aeruginosa that I highlighted before. So this is a cross-sectional study. We hope to enroll 300 patients, and these will be people with CF 16 years or older. So we're really targeting that kind of older children, adult age. This will be enrolling in approximately 10 sites. And because we need that sputum as the gold standard, we really are targeting expectorated, so those who are likely to give a sputum or at least induce sputum during the study visits. And we will be enrolling in different cohorts, including individuals who are in stable lung health or in pulmonary exacerbation. So we're going to be leveraging a clinic visit in which data will be collected, particularly like very detailed microbiologic history and concomitant medications such as antimicrobials. Then a clinical sputum culture will be collected and those data inputted. We will also be doing some clinical assessments listed here, and of course those novel specimen types: exhaled breath, blood collection, voided urine, and also saliva. So we are partnering with Alstone to have a separate Alstone sub-cohort breath study, which really the goal is proof of concept. We're going to be identifying the target volatile organic compound profiles for Pseudomonas aeruginosa. So we're really looking for Pseudomonas chronically infected folks and those who are non-PA infected. And the hope is to then, after we identify that, go on and expand to other disease states, such as, measuring treatment response or other gram-negative bacteria. So in summary, there's a lot of promising diagnostic technologies that are not based on sputum on the horizon. And what SenCF intends to do is really kind of jumpstart that work and expedite that work to our novel detection or infection detection diagnostics in CF, with the ultimate goal to detect early and treat to minimize that impact of chronic infection in the CF population. Special thanks to Chris for some of the slide content and the rest of the SenCF team. And that's it. I'm happy to take questions. Okay. Do you want me to take questions now or later? Oh, okay. Excuse me? So we've already done site solicitation and selected the early sites, but depending on the course of the study, it may expand. Yeah. Yeah. So there's 2 devices that we're going to be using for the SenCF study, one of which is approximately 5 to 15 minutes, and the second device, which I didn't show in the slides, which is like actually a little bit more experimental, is 5 minutes. So it's actually quite quick. But there's other breath collection methods that have been used by investigative groups that may take a little bit longer, but with these devices that we're going to use from 5 to 15 minutes. Yeah.
Is there a reason why you use the specific tubes for the exhaled breath condensation?
So I will say is that I hope I'm answering this question correctly, but sorbent tubes, it's a little bit more stable than some of the other modalities like Teplar bags. And I think that that's why we are doing it. I didn't say, but Alstone Medical is actually based over in Cambridge, UK. And so we have to kind of quickly transfer those over there, but they have to go across the Atlantic. So I think that that's the reason why that company particularly uses the sorbent tubes, which I think is gaining popularity for breath sampling. Yes.
I just wanted to make a comment, not really a question. So my son has CF, and this would have been great because at one point his cough had, started to increase, and that was in November. And it wasn't until even the end of May did he get diagnosed with like MRSA. And so this whole time he missed a lot of school and was in, just a lot of not sleeping, body aches just from the coughing. And we kept taking him back, but it was so far down in the lungs that, they didn't get it to the finding board. So just wanted to read that down.
Yeah, thanks for sharing that. That's why these are so important. Can be really challenging, especially in children or even right now, adults who are not readily expectorating sputum. If we can't get that sample, then we can't accurately detect, but it's still going on and they're still experiencing that clinical impact. So, we're hopeful that, I think it's very early stages, but we're very hopeful that, these modalities can aid in that sort of situation where infection can be detected early and then treated early so that people with CF don't have to experience what your son has experienced. Thanks for sharing that.
Gina, I have a question.
Yeah.
This isn't working, but I know it's very early, but do you envision any of these samples being able to be collected at home and then sent to a center? I know that we'd probably have to do another study to verify, but.
No, it's a great question. So I think, let me comment on voided urine. Voided urine, I mean, it can be very stable, especially if one could put it, stick it on the refrigerator. I mean, I think that that's definitely something that is I wouldn't even say it's a pie in the sky, but something down in the pipeline. But even breath collection, I know that there's investigators in British Columbia that were actually trying to explore home breath collection. And I think that these technologies, I mean, right now, the Receiva device that we're going to be using with our partnership with Alstone is quite involved, but I think the goal for the company eventually is to extend that to permit point of care. So whether it be in the clinic or at home, I think that there is potential for that. Yeah.
Great. Justin?
Are you guys doing semi-drops and some of these?
So no, yeah. So we're not doing bronchoalveolar lavage. We are using sputum, which we recognize is still a bit of a limited gold standard, but that's what we're using, sputum and induced sputum as really the gold standard. Yeah.
Oh,
What's the reason for using expectorate sputum versus
Yeah. So the only reason was it would be easier. It was because we wanted to leverage the clinical visits. And so the goal is still just proof of concept and banking. So although obviously these novel technologies are supposed to extend to those who can't make sputum, but at this stage, we really are trying to target those who are expectorators. And so that's why we had generated the protocol to involve really that expectorated sputum, but still incorporated if they weren't able to make sputum at that visit, that induced sputum would be obtained. So it's really just ease, cost, and trying to make it kind of embedded in that clinical visit.
All right, just one more.
Do you think some of the doses detect on self-limited infection because symptoms of the function on specifically like this?
So great question. I think that we don't know. I mean, the hope is that it could. I think. Oh, sorry. The question was whether or not that breath can be used to detect VOC profiles for subclinical, before the lung function decline, before the clinical consequences, the infection or whatever disease state. I think we don't know. I mean, that would be the goal. So, right now this is, for example, the goal is even just to detect chronic PA versus no PA right now, but wouldn't it be great to look at incident PA? So I think that we just don't know, but that's the hope of this technology, is just really too early to know. All right, thank you.
Thank you. I might go down.
Our next speaker is Dr. Manu Jain. Dr. Jain is a professor of medicine at Northwestern in Chicago. Go Cubs. He's a co-center director there at Northwestern as part of the Lurie Children's Hospital. Please welcome Dr. Jain.
Good morning. Thank you to the moderators for this kind invitation to present our work. It's titled Recoup 001, Remote Monitoring of Sweat chloride and exercise in people with CF. And before I go too much further, I want to do a shout out to my research manager who actually should be giving this talk. But for good reason, she was unable to come today, but so I'm here in her stead. But she was very instrumental in doing the study, completing it, and putting this presentation together. So I want to acknowledge her. So sweat chloride has become an interesting topic. I mean, historically, it was used for diagnostic purposes, and you had to go to a sweat lab that was accredited. And so it was not necessarily the easiest thing to implement clinically, especially at adult hospitals. In this era of CFTR modulators, sweat chloride has become potentially another, besides a diagnostic tool, may have other utility as well. So we became interested in measuring sweat chloride in a more seamless, clinically relevant way a few years ago. We partnered with some bioengineers who have a company, it was an offshoot of Northwestern, who are focusing on wearable biotechnologies for medical monitoring. And that's sort of the genesis of how this work started. These are my disclosures. So I have three objectives today. I'm going to discuss the feasibility and the potential utility of this sweat chloride and volume collection device. It's a wearable device, and I'll show you what it looks like in a second. Discuss the potential role of remote health tools such as heart monitors and sweat chloride as potential clinical and research tools. And then talk a little bit at the end about what the challenges we experienced with respect to remote data collection and interpreting this for human subjects research. So this is a diagram of what the CF patch looks like. And there's two or three key things. It's administered as via adhesive to the skin, and that's the bottom layer. And then on top of that are two different layers that have microchannels in it. And these are open to the skin when you adhese it, allowing sweat to enter as you either exercise or use pilocarpine to induce sweat. And then there's a top layer to sort of keep it covered. And you can see, I'll show you a better picture here. So the patch that we used has two channels in it. The orange channel, parenthetically, each channel, microchannel, has reagents in it that changes color when sweat enters into it. So the orange channel measures volume. And so each row is about 15 microliters. You can use that to calculate how much sweat is actually generated during a exercise session, help you to calculate how much volume needs to be repleted after you exercise. If you look at the purple channel on the bottom right of the patch, that is measuring sweat chloride, and it's a colorimetric assay. So the intensity of the purple color will be reflective of what the chloride concentration is. So that's sort of how the patch is designed, and you can see an illustration of how it's applied. So it's generally best applied on the volar arm. It's really important to get a very nice seal because you don't want sweat to be leaking around the patch. But we were able to show patients how to do this, and they get it's pretty easy to teach and for them to learn how to apply this patch. And after you exercise, this is a blow-up of what it looks like after you finish an exercise session. You see the orange telling you how much volume you've generated and the color of that. And in a perfect setting, you could actually, in real time, take a picture of that patch, compare it to a standard, and actually get a real-time measure what your sweat chloride is at that moment. So using this patch, we designed a study, a pilot and really a feasibility study, which had three different components to it. First was a pre-enrollment where we obtained remote consent, and we asked patients to keep medication and food diaries for a week prior to coming to the second part of the study, which is the enrollment visit. This was done in an exercise laboratory. And there were three things we did when they came in. It took about 4 hours to do the whole visit. The first thing we did was to do a paired collection using pilocarpine, one for macroduct chloridometry, which is a gold standard, and then the other arm was used to collect for our patch. We wanted to do a direct comparison between the gold standard and our patch to see how accurate this patch chloride was. So once they completed that part of the study, they got on an exercise bike and did a CPET where we had them exercise to the point of exhaustion, basically generating a VO2 peak. After they completed that, we let them rest, recover, hydrate, and then did a second exercise session at 50 to 70% of their VO2 max. And it's during this session that we actually collected the sweat for chloride measurement to see what the exercise sweat chloride would be. We theorized that people would be able to exercise for a prolonged period of time if they exercised at 50 to 70% of their VO2 max. So once they completed that, we collected the sweat, and we monitored this visually to make sure enough sweat was being collected. So this is an ongoing assessment in real time. Once they completed that and they were recovered, we gave them 5 to 6 sweat patches to take it home to complete the third part of the study, which was home exercise. We asked them to do 5 exercise sessions using the patch over a 2-week period. And then we also gave them a heart rate monitor to wear while they're exercising to see what heart rate they were able to generate and get to and maintain as a way to compare the intensity of their exercise at home relative to what we measured in the laboratory. So we enrolled healthy volunteers and people with CF. We consented 23 people with CF, and 20 actually completed the study. We can show you the some of the selected demographics. 55% of our enrollees were women. Their age was 21 to 63, so we really did the adult population. 50% were F508 del homozygotes. 95% were on a modulator, predominantly ETI. Mean BMI was almost 25. The mean FEV1 was 95%. Predicted. So, a pretty healthy population. A parenthetic, I should mention, we enrolled people who were self-described as regular exercisers. So we wanted to make sure that they were going to be able to complete the 5 exercise sessions. So we did bias it towards that. But it was a pilot study, wanted to see if this actually was accurate or not. So what did we find? So this is a graph of what the sweat chloride is when you compare our patch chloride to the gold standard, which is chloridometry, and this was using pilocarpine for induction. We found a pretty good correlation, an R value of 0.9, which was highly statistically significant. And then if you look at the graph on the right is a Bland-Altman plot, which is a way to assess whether the measurement is accurate at low, medium, or high levels. And what we found was that it was accurate whether you're measuring low chlorides, medium chlorides, or high sweat chloride. So it seemed to be not biased. The second graph here shows what is the relationship between the gold standard and exercise-induced sweat. The question was, does exercise replicate what you can measure using pilocarpine in terms of the chloride measurement? And we found that there was a pretty good correlation with exercise-induced sweat chloride. Remember, this was all happening in the exercise lab. And there was a correlation with an R value of 0.81. And again, the Bland-Altman plot indicated that it was accurate across a range of sweat chloride values. And then finally, we compared exercise compared to pilocarpine using a CF patch, just sort of an internal check of the CF patch to make sure it worked with both pilocarpine and with exercise. And again, the R value was good at 0.8, 0.79, and a good an absence of bias in terms of the measurement. So this was all done in the laboratory. But, the real value of this is, can you actually get people to do this at home? So what I'm showing you is data on the exercise sessions that people did at home. The graph on the left shows our population of healthy volunteers and people with CF. The people in green, or the bars in green, are people with CF on modulators. The one CF patient not on a modulator is in blue. You can see their sweat chloride was 100, which you would expect. And then you have healthy volunteers in orange. Interestingly, some of the healthy volunteers had sweat chlorides that were in the CF range, which has been described before. There are people without CF whose sweat chlorides can get into the indeterminate range. But the thing that I want to also point out is that the people with CF, predominantly on modulators, had a very wide distribution of their sweat chlorides, which is indicated by the bar. But then if you look at individual values, which are indicated by the blue, the black dots, those are the individual values. And if you look at certain individuals, they had a lot of variability in their sweat chloride on a day-to-day basis. And when we analyzed this was greater than we saw than in healthy volunteers. Then I'm graphing on the right is a comparison of the mean sweat chloride of all the exercise sessions at home with the exercise session that we did in the laboratory. And again, there was a pretty good correlation of 0.72, not quite as good because people were at home. And so we wondered if the poor correlation or the less good correlation was maybe related to something that was happening with people with CF. So we separately analyzed healthy volunteers and people with CF with respect to home and exercise, laboratory exercise, sweat chlorides. It turned out healthy volunteers correlation was quite strong, 0.92. With people with CF, their correlation was lower, was about 0.59, 0.6, consistent with the idea that they're having more day-to-day variability in their sweat chloride at home relative to what we measure in the exercise laboratory. The reasons for this we are not sure of. We have some hypotheses, but we don't know exactly why that is. So we did learn a few things along the way. It is not easy to do a study when patients are doing a big chunk of it at home. One of the challenges was how to assess whether a collection was successful or not. I'm showing you a picture of a successful collection on the left. The two in the middle and the right are examples of unsuccessful collections where if you look at the picture a little closely, the orange line is not fully full because it was leaked. You can see the outlines of the orange, but it leaked out, even though it looks like it was adhesed very well. And then on the right, that shows some leakage as well. You can see on the bottom right corner, it sort of lifted up as they were exercising. And then when they took the picture, there was shadow across the patch, which we had asked them to try to avoid that. That's not it doesn't kill the analysis, and we can still correct for that. But, those are issues that we did have to encounter. And then we also did a heart rate monitor during their exercise session. One of the things we asked them to do was to turn it on and turn on the Bluetooth component of this so they can communicate with a cloud so we could actually monitor their heart rate in a real-time setting. Sometimes they would remember, sometimes they wouldn't forget. That was not also a deal breaker because the device itself stores the information for the heart rate. So we could also download that at the end of the study when they returned the heart rate monitor back. So how do we sort of address some of these issues? Well, it required communication on a regular basis. We made sure we spoke to our research subject the day after they completed their first exercise to make sure they understood and felt comfortable applying the patch and the heart rate monitor. This ended up being like nearly daily communication between our research staff and our subjects. So the regular communication was really, really important for ongoing training. By the second or third time of using the patch, they became pretty facile with using it, and we saw that they were getting very comfortable. There were technical limitations with Bluetooth, especially if people were exercising outside, went for a run outside or a bike. It didn't always do a connect with the cloud as it collected heart rate monitoring, but like I said, we could collect it at the end of the study. We did build into the protocol that if somebody wanted to do an additional exercise session that was because one of the collections was a failure, we did allow that flexibility to do an exercise session, allowing us to collect more data. So this work is now published. I had the link there in one of my earlier slides. But the question now is, what are the next and future applications for this device? Well, we can envision using electrolyte and fluid recommendations for exercise sessions. You can measure it and actually get a real-time assessment of how much salt and water you need to take in to replete yourself. We've shown that a remote assessment of chloride is feasible. So does that allow you to monitor CFTR modulator dosing? adherence may also be something we could look at. Do we get more value out of generating individual sweat chloride profiles and assessing that to clinical response? You know, the way we look at things now is we get one measure after somebody starts a modulator in terms of a research setting. We can't do it clinically, but now we could potentially do it clinically and assess whether profiles give us more information as opposed to just a single measure. And then we are piloting obviously not every person with CF, especially young patients, won't be able to exercise to generate sweat. So we are piloting a patch that has pilocarpine embedded in it, so you could actually use that to induce sweat. So my conclusions are sweat chloride measured by a CF patch is accurate. Remote and recurrent sweat chloride measurement remotely are possible. Remote heart rate monitoring is feasible. We still need to do work to assess what the clinical utility of using this device to measure sweat chloride assessment remotely is. I want to thank my collaborators at Northwestern. You can see them there, and at Epicor, Ruz Ghaffari and AJ Arenosi, who helped to develop this technology. So thank you very much.
We talked a little bit about sweat variability and some potential hypothesis related to that. Can you talk a little bit more about that? And then also, would less variability perhaps mean better CF health?
Wow, okay. So like I said, I don't know why there's so much variability in people with CF. I can hypothesize, dosing in terms of when you dosed, you took your medication, and when you did the exercise. Maybe one potential mechanism. Whether you took fat with your last dose or didn't, that we know that impacts absorption of your modulators. How much salt you ate the night before, how well hydrated you are could also be playing a role. Are you on other medications which are inducing different metabolism? So I think these are all hypotheses that need to be tested. Maybe just forgot to take the dose the night before. So, I think these are all sort of interesting questions, but our design does not allow us to answer that question. And the second part was about sweat variability. Yeah, I think that, it kind of makes sense to me, although unproven, that the greater CFTR function recovery you have for a longer period of time is reflected by a lower sweat chloride. I think over the long term I would think that would lead to better long-term outcomes. Obviously, that's not been proven, but that would be my hypothesis.
I have a couple of questions. First, I love the study design, but with the heart rate monitoring, I'm curious, did that matter? Or is that like if somebody in one session, they hit 40 to 50% of their VO2 heart rate, another was 70 to 80, is it just like as long as they sweat, it was okay and the heart rate didn't matter? Could you simplify that in future designs or did that matter? So we wanted the heart rate data to assess how intense their exercise was at home relative to what we did in the laboratory. But in terms of sweat collection, it doesn't matter. You can exercise at 50% for an hour or 70% for 30 minutes. As long as you collect enough sweat, and you can assess that visually as the color is going down the tube, that's fine. Yeah, and the other part too is like I like the idea of protocols for hydration and electrolytes. And what about measurement? What are you thinking of like measurement, like whether it's urine specific gravity or lights or some other thing? Oh,
in terms of assessing whether
just to see like is there a correlation between can you measure their hydration status relative to their sweat?
Yeah. So I think that's a tough one, but yeah, I think urine, your specific gravity, osmolality potentially, also recovery from exercise if you hydrate better. Is your next exercise session easier? So there are some of the things we're looking at, but the first thing is just to see whether it impacts how much fluid you take, right? If you let people just drink and replete based on how they feel versus what's recommended by this patch, is it different? I would say it probably is different because I think there's data suggesting we don't hydrate and replete just based on symptoms as much as we should.
Thanks. I love the study.
Thanks.
Hi, Jonathan Zuckerman from Maine. Really interesting study. for those of us who like to exercise. So it's a lot of fun. A question about, since most smartphones have access to the weather conditions, I was wondering if you had any of that metadata about the humidity and temperature that the people were exercising under.
Oh, that's a great question. We do not, and we could potentially go back. We have the dates on which they did the exercise. That was captured, so we could go back and see if that was playing a role, sort of a multivariable analysis. That's a great idea.
Just a quick question about like the adhesive design for the patch. Like, do I use like a dual layer system or like a silicone adhesive?
I am not the right person to ask for that question. This was designed by my collaborators, but if you send me an email, I can find that information for you. But what I can tell you, it's very well tolerated. Patients, a couple of people got some mild erythema after they took it off, but really had minimal adverse events with wearing the patch.
And kind of like a follow-up with that. So like, you showed some of the pictures like that for patients who didn't have a good seal. Like, so is there some type of visual, like maybe suggestion, like is there a visual indicator, like a sensor light, or maybe like a mobile app that would say, hey, there's a sensor error or something like
Yeah.
that? Kind of like CGMs do that.
I see. No, we don't have that in the design, but I'll take it back to my colleagues and see if we can build that. But, we spend a fair amount of time in troubleshooting with our research subjects about making sure the adhesive, they adhesed it appropriately. But that's a great idea. That's some feedback to say, you're good to go would be, I think, a nice design implement. Thank you.
My question's really close to hers. Did you guys prep the site the same way you do for a regular sweat test with like alcohol? And did you shave the arm hair? Is that why that didn't?
If they need it, it's really key to get really as flat of an area on the arm as you can because you want it to adhere well. And we clean it like you suggested. And for some people, we did need to shave their arm. Generally not a lot of hair on the volar part of the arm, but some people do have it. So we'd have to do it sometimes.
And the one where the orange line it was all faded. Did you guys troubleshoot and figure out why that didn't work?
That was a home exercise session, so we couldn't do it.
Okay.
Thank you. Thank you.
Thank you very much. Okay, thank you again. We're going to pivot just a little bit, and we're going to ask our friends from UNC to come on up. Ellie and Julie are research coordinators from UNC Chapel Hill. Julie is somehow keeping her head above water with 5 kids, as an avid workout junkie and a dabbler in the New York Times word games. Ellie is working toward PA school and is an avid volleyball player and apparently takes a lot of pictures of food as the foodie of the group. So please come on up, Ellie and Julie.
Hi, everyone. As I said, I'm Julie Gowdy. I'm presenting with Ellie, my colleague, on an app that we developed as part of a remote care program at UNC, including other sites with USC, Maine, and Case Western. Here's a disclosure. I do receive the LEC award from Cystic Fibrosis Foundation. Thank you. So we wanted to explore the role of mobile health applications and how they could be integrated into the evolving CF care model. Our rationale for this was that with modulators, there's been an improved clinical outcome in our CF population. And in 2021, there was an article published in the Journal of Cystic Fibrosis saying that there's an increased desire among this population for telehealth and utilizing telehealth. They also have an increased experience with home spirometry, and so that could also be incorporated into a program. That being said, we do still know that CF complications exist, and there's a need for regular monitoring. In preparation for developing this remote care model, we send out a survey among the sites to see the perceptions of remote CF care among our population. Our respondents said, 60% of our respondents said that telehealth was useful for well and sick visits, and their ideal combination would be about 50% in-person, 50% telehealth. So that was an important component we wanted to integrate into our care program. 58% said that forgetfulness was a primary barrier to getting home spirometry done, so having reminders throughout the care model was an important part as well. 76% said that collecting sputum in clinic was very difficult, but that home collection was very or somewhat easy. So we wanted to also include an option to bring sputum to clinic, send sputum to clinic, or have those analyses done locally at other labs. In addition to that, 53% actually preferred doing annual labs and OGT collection at their local labs to ease the burden of like coming all the way into clinic to do that. So that was also important to have localized options. But overall, 86% were very or somewhat interested in a remote CF care program. Out of that, we developed the CF at Home app, logo designed by Scott Donaldson. As we evaluated our participants in this program, our potential participants in the program, we wanted to find clinically stable adults, those that providers felt were good candidates for moving to about 50% telehealth routine visits. We wanted to find people that were able and willing to use an app on their mobile device, and also able and willing to use a home spirometer and body weight scale, which if they didn't have that already, we did provide the home spirometer and the scale. We didn't actually take into consideration their actual lung function or modulator use. It was just an overall clinically stable status. The program components then included home spirometry and home health, or home weight. Then performance of annual labs, OGTT and DEXA were taken into account. And then their sputum cultures, how many they got throughout the study. And then surveys were administered at 6 months and 12 months. Compensation was given at enrollment of the program and then paid for every survey completion they did. So when we enrolled the patients, they would download the app. It was available in Android and iPhone app stores. They would create an account using a dummy Gmail account so that we didn't collect any unnecessary PHI. And after that, this was the platform that the RCs would use. We would assign a pre-curated schedule of like your general like monthly home spirometry, monthly home weight, and then we would tailor in when their annual labs were due. And the idea was mostly just to set it and forget it, to let the app do its work. You can see up here, this is their first set of cards at enrollment. So it tells them the rules of engagement, gives an overview of what they're going to be expecting in the program, and then home PFTs, sputum culture, and how payments would be done. So when the RC would schedule a card to be sent, the user would then receive a notification on their phone, like you can see here, of, hi, I've added something to your CF at Home app. The idea was then tap into it, and it would pull up a card. Our example here is for the home spirometry. As you can see, there's an education component of it, how, why, when to use to do the home spirometry, like best practices. Furthermore, we included a video on how to use the Zephyrix app for their home spirometer. And then the idea was to report your FEV1 monthly so they could track it, as well as indicate how are you feeling today, so that if there was any sort of, we would collect a baseline FEV1, and if there was a drop to a certain threshold, the RC would then alert the nurse coordinator to reach out to that patient to check on them. Also, we reminded them that you can reach out to the clinic if you are feeling unwell. So we provided contact information for the nurses and a link to go to UNC MyChart. And with that, I will turn over to Ellie to find what we found.
All right. Hi, everyone. So we're first going to discuss some of the quantitative data we got from our surveys, and then also discuss the experience with the app from the RC perspective. All right, so like I said, we're going to talk first about some survey findings. So we sent out a satisfaction survey through the app, and overall we got 95 responses. So just to orient you a little bit to the data we'll be looking at the next couple slides, so you'll have the question we asked on the left-hand side with the legend at the bottom, and then percentages in the boxes. All right, so as Julie mentioned, we only sent out reminders monthly, and we bundled these to make sure there wasn't app reminder and app fatigue. However, 44% found that those reminders were a little too few or far too few. And this obviously affects engagement and participation with the program, and that you can see this later in the data that we talk about. All right, so in terms of what went well, 80% found that the app was easy to use. Around 70% found that the reminders were useful. About 2/3 found the information and education materials helpful. About the same percentage found that home spirometry improved their telehealth visits. About 60% reported they had to take fewer trips to clinic. And about 50% reported remote care was as good as in-person visits. So to talk about what could be improved, these green boxes on the right-hand side are not applicable or haven't tried to do this. So 50% or greater disliked or didn't even take advantage of local options that we offered, such as home sputum collection, arranging local OGTTs, and local DEXA scans. However, 42% reported that they were more likely to get screenings due to the app, which just means that these reminders worked well for in-clinic visits, but not quite as well for local opportunities. All right, so we also wanted to investigate participant engagement within the app. So this chart shows that the percent response to the first card, and which simply asks, do you agree to the rules of the app and the program? So in the blue, you'll see those that agreed to the card, and then in the red, those who didn't even respond. So the width is proportional to the number enrolled at each site, with UNC enrolling 60 participants and the other 3 sites enrolling roughly 25 participants. So as you can see, Maine had much higher engagement with this very first card. Emma, the RC at that site, attributes their success to her continuous engagement with patients in clinic. She was able to check in on them and ask how they were doing with the app and troubleshoot any issues. She also cited that their clinic was able to implement a new home sputum collection process, and the patients really responded well to that and gave her more opportunities to have FaceTime with those participants. This also shows that while we all can use the same technology, results are very approach dependent. All right, so this next figure illustrates how the very first card is a marker for engagement throughout the entire program. So on the y-axis, you have home weight per month, and then each individual bar of the histogram is an individual participant in the program. So on the left-hand side, you have the 64% that responded to the very first card, and you can see that overall they had much higher home weight frequency input, and there was a larger range of frequency as well. However, on the right side, the 36 that failed to respond to card 1, they had much lower home weight frequency, with 57% never entering weight data at all. So this just means that early participation is very key to sustained engagement through a home remote CF program. All right, so to get into some things from the research coordinator perspective, there were a lot of unanticipated technical issues, and it was not quite as a hands-off process as we had hoped. As Julie mentioned earlier, we couldn't have PHI in our app, so we had to use a dummy email, which the participants couldn't access themselves. So this caused a lot of logouts and forgotten passwords that the RCs then had to troubleshoot themselves, and it often took multiple rounds of calls and communication for that to be fixed. The platform we used was quite easy to manipulate, but the Nudge Coach provider went out of business early, so not all of our participants had the opportunity to complete a full year in the program. There also was data discrepancies on the dashboard of Nudge app versus the printed out report that we created at the end of the study. So in summary, the app and the program were very well received. Participants found the app easy to use, and patients liked the ability to be involved in care and track their own data. Engagement wasn't as high as we hoped, and we need to find the right reminder frequency. As shown earlier, that monthly was too few for the participants in our study. We also need to solve IT challenges and identify mobile health partners to help reduce the burden on research coordinators, and to also potentially work with established platforms with maybe addition to MyChart, because a lot of participants reported not even wanting to download an additional app. We're currently working on additional analyses to demonstrate if this kind of program affects outcomes. But we are super excited about by the idea of a program like this and furthering the CF care model and giving patients a better way to conduct remote care. All right, we want to thank again our collaborators at Maine, USC, and Case Western, and thank you guys for listening today. All right.
Do you mind going to the microphone? It's much easier for all of us to hear you. Thank you.
Hi, I'm Rachel. I'm a project manager based over in Ireland, and we're looking to set up an app to measure adherence in our patients. I'm just wondering, what was the rough cost of the software development?
Okay, we'll let our PI take that one.
Yeah, so this was actually surprisingly cheap, I would say. It's not an option now because the company went out of business, but essentially when we were conceptualizing the project, we kind of looked around to find what would be a coaching application. It cost $11,000 per year for up to, I think, 300 participants. So, you know,
Yeah,
not too
cheap
much.
enough.
A few bucks.
And then just one other question. Did you engage with any patient or public involvement groups prior to developing the app?
Can you repeat that? I'm sorry.
Did you engage with the CF community to see if this was an app?
Yes. Yeah. So the initial survey that they described was before the app was developed. So we got their input. They seemed to be enthusiasm for it, and we tried to incorporate the components that they were asking for.
Brilliant. Thanks a million.
Thank you.
Hi, Dennis Kajaliadis from Penn. Can I ask you, how did you approach, patients? Was it basically mostly an unsolicited, group? Did you select people that were more likely to participate? This is great. This is a great opportunity for patients that want to engage, but the usual, thing is usually we have some patients that engage with all those things and other ones that are impossible probably to catch. And it's how we deal with that.
It was unsolicited. We would screen who was coming to clinic. So we approached them in clinic, and we would check in with the providers first to make sure they were people that were eligible, and that provider felt comfortable not seeing them as frequently in person. And we did have the limitation too, where we needed someone that was within the state in order to be eligible for telehealth visits as well.
But it sounds like this is at least a relatively wide selection, so it's not surprising that you had some trouble with engagement in some perspectives.
Yeah.
Hi, I'm Emma McWilliams. I'm from Boston Children's, and I just wanted to know if you had ideas about any other engagement that you might look at. Is there an opportunity that you could look and see what they click on when they get in the app, things like that?
Nudge Coach didn't have the capabilities to see how they interacted exactly. The biggest points of interaction were when they had the fillable fields.
Yep.
That was the data was reported to us. We could see if they opened the card.
Yeah.
It had a little eye if it if they actually interacted with it. But that was also another difficulty with the platform itself, because if it was a regularly occurring card, it didn't always show the exact, like, when it was sent, when it was opened. So there were some hiccups there. But otherwise, we couldn't see, like, where they were interacting, if they were scrolling through the whole card or not.
Yeah, so you couldn't see if they like opened the app but didn't click on anything in it and then closed it out like that. Okay.
I'd want to know that as research coordinators helping to potentially design an app, what are some of the tips that you would give to the app developers to say, this is what you need to do to make it feasible for people to do, and then also reduce some of the touch points between the coordinator, the people on the other side working with the patients.
I can take the first part and you might follow up. So I wasn't quite there for the whole app development, but we did hear a lot of feedback about the notifications not syncing up with their phone as easily. So having preset like systems in place for that. If you have anything to add.
I know it was, I think it would be easier to get buy-in from also the clinic members. Some of them were requesting, hey, could you actually send them a notification to schedule their in-clinic visit?
Right.
Which was not the point of the program.
Right.
Which is why we kind of thought integration into something that's already there, like UNC, it's like my chart.
Right.
If we could find something that would fit that. The other problem too, people just don't want another app.
Right.
But potentially, if it was more of a like big guns organization, like if the CF Foundation was able to kind of come up with something where it was a more official,
Not so homegrown kind of, but I
a
don't mean
little
that.
bit,
Yeah.
less likely to go out of business.
Yeah.
So, but there were the people that were most engaged were the ones that were already on top of their health. They're the
Right.
ones that had the notebooks, and the one that I don't know, we had one participant that she sent in a sputum culture within the first month, and but she was on top of all of her stuff. And I think for those people having and expanding a little bit more on tracking like FEV1 or other parts of their health, I think would be helpful.
Right.
I don't know how to engage the people that don't want the app.
Right. Yeah, I just find with all of with remote things, as easy as it's supposed to be for the research coordinators, we always end up spending a lot more time than anyone expected trying to get the data and keep people within windows and all of that.
I think also enabling
Engaged,
a third party being able to do that, the tech support.
Yeah.
Too.
Oh, yeah, tech support would be great. Yeah.
We discussed that there might be a potential for an AI part of an app,
Yeah.
less risky, just to be able to troubleshoot the technical parts of the app. I don't know.
Right. As long as they're not hallucinating, right?
Yes.
I'm Emma from the main site. I can actually talk a little bit about what would be helpful as well, because we had some great engagement and people that wanted to be engaged, but the app itself, how it was set up, if you click the notification, we could make sure that everybody allowed notifications. That's the first thing. We'd send them out and make sure they could see them. But if they click on them, as soon as they go into the app, that notification goes away and there's nothing that tells them like on the icon that they still have a notification. So they could go in and read it and be like, oh, I want to do my spirometry, but I'm not home right now and I need to run home.
Yeah.
And they would forget. So that is kind of like one of the biggest things. And also they mentioned it, but being logged out, if there was like a Face ID or thumbprint or whatever, being able to log in a lot easier. So just the overall notifications, because we had some really like awesome participants that were like, it's really frustrating because I can't remember because they go to read it, as like the biggest things. And then to have less RC burden, working with Clinic, every single site used Clinic in a different way, which was really cool, just totally based on their site. But like they kind of noted, this was the first time we used a remote sputum process with our clinic. So it was super helpful, and we actually used it for some sick visits. And we got samples from people that we hadn't got samples from since like 2019. And we're able to see like bugs that they grew and everything. So it was really helpful for our site. And I think that's also what made it like what people wanted. And they definitely made it their own experience. But I would say the notifications and how to figure out how to combine it with clinic is the best part.
Thank you. Great. Thank you.
Thank you.
Okay, so our last speaker is Dr. Rachel Salyer, who is the research coordinator for the PRIDE CF study at the WVU School of Medicine. They have expertise in mixed methods health services research and have experienced the novel challenges faced by researchers conducting online research recruitment in the age of AI bots.
Good morning. My name is Rachel Salyer. I use she, they pronouns, and I'm a research coordinator and postdoc at the Gender Research Lab at the West Virginia University School of Medicine. I do not have any disclosures, and this is the CFF's disclaimer. During my presentation today, I hope to help you identify current challenges related to bot responses in online survey data collection, examine historical and modern efforts to protect and clean survey data, and develop strategies for project-specific data protection and security in the current research environment. Bots are software programs that imitate the behavior of a human, as in participating in a chat or performing automated tasks on the internet. These programs are often used to help humans complete tasks and find answers via search engines more efficiently. But as we've seen in the news in the past few years, bots have posed a continual challenge in healthcare, social media, and even research data collection and dissemination. To ground us in everything we're experiencing, this is your reminder that ChatGPT was launched roughly 3 years ago. I suspect many in the audience have used ChatGPT or a similar AI platform for a wide range of tasks. I would love to know more about what you have used these platforms for. Here's a QR code so you can anonymously share. I'll have a team member summarize your responses, and we can follow up on those in a bit. The QR code will be on my next few slides, so you can continue to access it. According to a recent study of ChatGPT, as of July 2025, 18 billion messages were being sent each week by 700 million users, which represents around 10% of the global adult population. These researchers also state that within work usage, we find that users currently appear to derive value from using ChatGPT as an advisor or research assistant, not just a technology that performs job tasks directly. According to an MIT News article released at the beginning of the year, each time a model is used, perhaps by an individual asking ChatGPT to summarize an email, the computing hardware that performs those operations consumes energy. Researchers have estimated that a ChatGPT query consumes about 5 times more electricity than a simple web search. While these programs absolutely can be helpful in so many ways, they can also be helpful to those with disruptive intentions. One area that has experienced significant disruption is online survey research. These articles are just some of what's been published in the last year. Many researchers are working hard to identify research challenges posed by bots and develop strategies to protect the integrity of data, especially data collected via online survey. Here we can see responses to an interest survey with the question prompt, are you currently seen by a CF care team? If so, where? If not, at what clinic did you last receive care? For those of you that can read this, any guesses as to which responses came from bots?
answer was, I can't remember.
Yep.
What was
Yeah, so these are the ones that jumped out to me, right? I decided not to see my CF clinic because of my work. While that I'm sure does come up from a visit to visit standpoint, I find it hard to believe that someone has never established in any capacity. And then I can't remember, as some of you pointed out. How about here?
What was it?
Med Express. Yeah. Yeah, so the ones that stood out to me here were Med Express, Game Day Men's Health, and Alexandra. So what you see here is a non-exhaustive list of some of the proposed approaches to managing the presence of bots in online survey research. Some of these prevent survey completion, or at the very least can flag possible fraudulent or bot completion for researcher review. Completely automated public Turing tests to tell computers and humans apart, or CAPTCHA, can be implemented using both Qualtrics and REDCap survey software. This test was developed in the late 1990s by Carnegie Mellon University. Yahoo was the first to implement the use of CAPTCHAs in 2001 to combat spam bots. Use of CAPTCHA can act in a prevention capacity to reduce the likelihood of bots accessing the survey. Both Qualtrics and REDCap use Google's reCAPTCHA, which presents some image-related challenge that is easy for humans but challenging for bots, or the simple "I am not a robot" checkbox. Google has now introduced reCAPTCHA v3 to move beyond one-time verification for some cases. This new reCAPTCHA uses information like mouse movements, browsing history, and browser fingerprints to assign a risk score without interrupting users. This score will then help you determine whether a user is a human or a bot. If you want to use v3 in Qualtrics, you can simply turn on the bot detection feature. REDCap does not yet use v3. Honeypot questions are hidden question traps for bots embedded in surveys. These questions are undetectable by the human eye, but viewable by bots. These are easy to embed in a REDCap survey with the @hidden_survey action tag seen here. These two questions shown here can only be seen by a bot, so bots completing the survey will respond to them. Humans will not see either of these questions appear in their question list when they're completing the survey. You can also set question completion alerts so that a team member can be notified immediately upon completion of a honeypot question. Seen here on the right, this will allow immediate research team member notification upon the completion, as opposed to detecting this in the data inspection phase of analysis. IP address tracking can be done in both REDCap and in Qualtrics, as well as numerous other platforms. In REDCap, IP addresses are captured through the use of the electronic consent framework. Within Qualtrics, this data is collected automatically unless the researcher opts out of this data collection. Geolocating also provides geographical insight into where participants are completing the survey. This information, along with repetition and IP addresses, can also be indicative of fraudulent responses. It is important to note that virtual private networks, or VPNs, encrypt your internet activity and mask IP addresses with one from a nearby server. In this way, VPNs obscure specific locations of devices being used and broaden internet activity to a VPN server. While this is helpful for researchers who do not want their data and activity intercepted, it can also obscure the IP addresses of those completing online surveys. While more time-consuming and tedious, establishing a consistent schedule of team member record review can help to catch bots before incentives are dispersed or fraudulent data is analyzed. Individual records can be reviewed in REDCap, where completion time is also displayed, as seen here. If completion times are too short, the survey response is suspicious. Upon manual inspection, if responses are suspicious, you can contact participants via email to set up a verification phone call. Sometimes these emails are not returned, but when they are, you can ask project and participant-specific questions to verify identity. Questions can be broader, like, how did you hear about the study? where are you currently living, or more specific, like what center do you receive your CF care from, and who is your primary doctor. Manual verification can and should be done whenever time to completion, IP addresses, or geolocation info seem suspicious. Use of open-ended survey questions also creates space for research team member detection of bots via manual inspection. When responses read as incomplete sentences or incoherent buzzwords without meaning, that is a clear indication of a bot response. Here we can see 3 columns of data. All are actual responses to an open-ended survey item. What advice do you have for other parents? Any guesses as to which of these 3 were bot-generated responses? So the left column is likely real parents, whereas the middle is most certainly bots from the pre-ChatGPT era. What I find fascinating is that the right is very likely bot responses from the post-ChatGPT era. Despite more robust measures for safeguarding data collection, in fact, all the measures I previously described were applied, bot responses still infiltrated this survey, and we are now in a time where bot responses look a whole lot like human responses. Regular data inspection and thoughtful recruitment approaches are critical given the current research landscape. There are a variety of additional approaches that you can use to help safeguard your online surveys. You can consider single invite recruitment or a closed invitation list. There are machine learning algorithms too. You can also create duplicate surveys based on recruitment avenues so that if any links are compromised, you do not have to withdraw the entire recruitment effort. It is also time to seriously consider working with survey research companies or develop regional or national opt-in survey networks of our patients and their families where participants are pre-vetted and individually invited to safeguard against these data bot swarms. There are a variety of ways to attempt to safeguard your online surveys from bots and also detect bots within your surveyed data. Using a combination of the methods I discussed here today increases the likelihood that you'll be able to have confidence in the integrity of your data, though there are no guarantees. With the risk bots pose to online survey research, researchers need to consider the additional time it will take to manually inspect data throughout the collection process and to avoid relying on conventional strategies in the new AI era to more safely address their research questions. Thank you so much for your time and attention. I want to thank the amazing lab that I get to be a part of. I could not do this work without the support of my boss and the efforts of our medical student mentees. I'm going to briefly share some of your responses to the QR code questions because my boss was kind enough to summarize those and bring them over. And then I'm happy to take any of your questions. 97% of you said yes to use of AI platforms. Wow, 24% are worried data is impacted by bots. Okay. 56 of you aren't. I need to talk to those 56. How you've used it, coding, writing, all right. Workout programs, proofreading, simplify patient-facing language. Scripts for organizing data, write IRB summaries, grocery lists, meal plans, travel. I think that says homework. Scientific searches, make slogans. Interesting. Pick a house. For writing emails, I will admit I have used it for that purpose. See what I look like with bangs. It's important to do that beforehand, you know. All right. And how it's impacted. Environmental impact is worrying. I worry in general of misinformation and confidentiality. Yeah. No, all of this, honestly, yeah, I get it. Any additional questions?
I just want to comment that the 56% of you that think your data's never been impacted by bots are wrong.
I have a question.
Yeah.
Can you talk a little bit more about these, like a pre-vetted survey population? And what would something like that look like maybe in the CF space?
Yeah. So something in terms of like a pre-vetted like group of participants, I think honestly, in my mind, that's something the community voice could be hugely impactful with. I mean, they've already been really helpful for us with a lot of our survey data collection, right? Because they have direct contact with the community. So just kind of creating a participant pool, especially if the CFF was able to do that in collaboration with community voice, I think would be huge just for all CF researchers who were interested in working with the population. But then beyond that, just continuing to grow and expand the network.
I think I heard the other night you guys had some bot challenges with an ongoing study right now in the CF space. Is that accurate, or what study was that?
Yeah. I mean, so multiple team members, like relative to us. I mean, I would say, so there are multiple different like survey projects that I work on currently. A lot of it, at least within the context of Pride CF, we've been able to, it wasn't so much that we were concerned about bots, we were concerned about people possibly misrepresenting themselves and getting a hold of our interest survey link and kind of reaching out. So we did a couple verification phone calls, or at least sent out numerous emails to schedule verification phone calls, most of which were not returned. But then it's just a matter of getting on the phone with someone or getting on a Zoom with someone and then verifying their identity. And in some cases you have to take a few more steps based upon the answers that they provide you or just the concerning responses that they're providing in that intake relative to the demographics they're using. But yeah, I mean, it's an ongoing problem and it's something we're always thinking about. I mean, the first thing that we're doing as soon as data is coming in, if anything that I'm just reviewing as it comes in looks even slightly questionable, I'm taking it back to my boss and having a conversation immediately to figure out what our next steps are as a team and what we can do to verify participant identity. But then also at the same time, we don't want to, make anyone who is a real individual completing the survey, we don't want to put them in a position where they feel, pushed out or not validated in the fact that they are reaching out and they are trying to participate. And we just, again, continuously remind people that this is just to protect the integrity of our data, to protect the integrity of our research. And it's with the interest of the population we're researching in mind.
I think that you were at the mic first, so go ahead.
Oh, yeah. Two things. I'm the one that said homework. To clarify, I was remembering how to do long division for my daughter so I could help her. So there's that.
That's fair. That's fair.
Secondly, this is very technical and I don't know if you can answer it, but it's just like the curiosity is killing me. Like the honey, I think you called it the honeypot responses that only the bots are able to see. Is there like a low level explanation of how that is?
I don't know how. Okay. You might know how, actually.
In REDCap, there's action tags. That's what you want to look up, is how to use action tags in REDCap. So you can apply different behaviors to so you write a question in REDCap like you normally would, and then you add this action tag that makes it hidden in survey mode. And for some reason, so a participant won't see it, that question's invisible, but because of the way the bots interact with surveys, they will see it and answer it. But it's action tags or wait, they're very cool. You can do a lot of stuff with action tags.
So that screenshot that I included was like on the back end. So like you see it as the researcher designing the survey, but like when you actually publish the survey and push it out and someone clicks the link, that will not show up to any of you.
And the survey just knows because of, again, the way the bots infiltrating the system.
Oh. They said that sometimes the text is white on white. So we wouldn't be able to see it, but the bots.
Thank you.
Super interesting.
Just had a question about, well, first, great talk, great topic. Just had a question about how do you as researchers balance this issue though? Like if you want to do, if pre-vetting and trying to look at a particular population for these, survey studies is a way to circumvent these bots, then you're at risk for other things like bias that is introduced in your study. So how do you balance that? And is it that you would recommend other researchers who are doing this to use those other kind of potential solutions, such as the honeypot questions and captions, stuff like that? Would you recommend those over the pre-vetted and things like that? So can you just comment on that?
Yeah, I wouldn't. I mean, this is a combined strategy situation, right? This isn't just try one thing and hope it works. And one of the other things that I will say is that what I am very fortunate, like with the team that we work with and with the collaboration we've done with Community Voice, is we work very closely and do a lot of community engaged research. So we are directly in contact with a lot of the population that we're researching. So in that capacity, going that direction and making sure you're staying as close to the population that you're trying to survey as possible is like first and foremost, like step number one. And then beyond that, working with a lot of these companies, or like what I suggested, like, what would this look like in terms of pre-vetting and creating a participant pool? I mean, that's no different than, in my mind, the current participant pool of like any patient with CF that's participating in research studies regularly. And there are a lot of those CF patients that are regularly participating in research studies. So it would just be a matter of right now, like we're just doing the work to try to find them. Whereas like thinking bigger, broader, the CFF knows them and knows where they are. And especially Community Voice is connected with a lot of patients with CF. So in my mind, that is what that would look like in combination with all of these strategies that you would also want to employ just to protect from, again, outside people with poor intentions gaining access to your survey link. And the targeted recruitment I kind of talked about too is, again, that's what I say, like we've partnered with Community Voice for numerous times in which we've recruited, and they do a really good job of, they know who their people are and they disseminate the survey link accordingly. And we have not had issues with potential bots when we have worked and disseminated our survey link with Community Voice participants. That has never been an issue with like bot infiltration in that targeted recruitment. So just kind of being intentional with where you're going and when you're recruiting, and that was creating sometimes duplicate survey links to collect data so that you can get a better idea of like where those bots could be coming from.
Yeah, just in response, because Community Voice, I agree, it's just a great resource, but those are like the most engaged. And so those responses that you may get from those incredibly engaged may not be representative of the entire CF population. That's what it's kind of just hard to balance.
Of course. Yep. No, I completely agree.
Hey, I'm Caroline from UNC. This may be kind of ironic, but have you ever considered using AI to detect the bots? It seems like it worked.
So I'll be the first to say that like I don't trust a lot of it compared to like myself and my interpretation and my judgment. So I am a skeptical user of AI and ChatGPT. I have not fully adopted use of ChatGPT. So no, I have not considered doing that. I would be interested if someone has. Have you? No. I just have a lot of concerns, honestly. And I still think that like ultimately it's going to come down to us as humans, us as research coordinators, us as PIs, doing the work and reviewing these responses. I think a lot of this has come out of, how can we do it faster? How can we get more done? How can we be more efficient? And I think that rush is what has got us to this point. So I think there's a point where like you do need to build in, like we do need to consider and build in time into, any of these research projects to make sure we are reviewing data, and there is time to do so. And sometimes I think slowing down and taking a second to check things out is going to get us farther in the long run than trying to just find ways to do it faster with AI.
That's a great answer. A bot could never come up with that. Thank you.
I'm about to show my deep lack of understanding of tech, but what is the point of bots doing this? Is someone twisting their mustache somewhere and going, I've released bots and I'm going to mess with this survey? Are they gathering data? What's happening?
So it depends, right? Essentially, yes, in a lot of capacities, these are people with poor intentions doing things that you and I could never fathom because that's not what we want to do. Right? Like it's just a different medium in which people have found to, con individuals. And
That's the upside, just
I
messing with
mean,
people somewhere.
if they can make their way through, they could probably get a decent amount of money, depending on like money that's coming out of these surveys, like from an incentive dispersion standpoint. I don't ultimately know the answer to that as someone who's just trying to do good. That's like, these have never been my goals. So just messing with things for messing with things is just not in my nature. But I can't really speak to their intentions aside from, in some of these, surveys, there are monetary like incentives, right? So there is financial incentive to completing some of the surveys. And if you find a way to get a bunch of bots through and complete a bunch of surveys really fast and get a bunch of money, I guess that's incentive enough. But I mean, it's just like everybody else who lurks on the dark web and everything else that like, I don't go down that rabbit hole. I don't know what people do in those circles. So
People have too much time on their hands.
There's a lot of money to be made, and that's a big part of it, right? So I have used machine learning and mixed language learning models to try to find the bots, and it becomes a predictive algorithm, right? And so I'm, 86% confident based on my gold standard, which is all this other stuff, and my community partners saying that sounds like a bot, not a parent, right? But it's not perfect. And so the best way to protect your data is to prevent the bots from getting into your data. So yes, the community voice population is going to be different than if you put a post on Facebook for anyone whose Facebook profile indicated they might have CF. But the chances of you finding real humans are very, very low with that Facebook post link, especially if you advertise there's any money associated. I've had colleagues that posted on various forums and have come to me personally and said, somehow our Qualtrics link had 50 participants before we launched the Qualtrics link. So how is that possible, right? I've had 7,000 responses in an hour before. How is that possible? So I think we need to be really thoughtful, and I wonder sincerely how many published pieces are out in the literature that are fully hallucinated. We've also had people who have found that they go to do an interview with someone on Zoom, and that person is not anywhere near where they said they were. They are not anywhere close to the likely age they said they were, right? And so people do these things because it's income, especially in potentially other countries. You can jump on a Zoom link anywhere if you get access to it. So we need to be mindful that the data we think we're collecting is not always what it appears to be.
Hi, I just wanted to let you know that I did ask ChatGPT if they can detect bots, and they said to a degree, so.
And that is, I feel like that's such a good response to close this session out on, Cool. Thank you all so much.
Everybody.